Good day, everyone. We are the Group 4, and our topic is gene editing, gene gun, and alternative techniques. Hope you guys learn a lot and enjoy!
Good morning. Hello there. I am Agendo Randy Nabakita, and I am here to discuss with you the history of genetic engineering. The history of gene editing began in 1976, when scientists first discovered how to cut and insert DNA segments using restriction enzyme. Remember those two words, how to cut and how to insert. So now, let me walk you through each scientist that started genetic engineering. In 1960s, Werner Arber at the University of Basel was the first to propose and demonstrate the bacteria have a natural defense mechanism. He theorized that bacteria produce restriction enzymes that cut foreign DNA while protecting their own DNA with a modification system. Arber's work established the concept of restriction and modification systems. He predicted that specific enzymes were responsible for this. Next up, in 1970, Hamilton O. Smith, working at Johns Hopkins University, isolated and identified the first type 2 restriction enzyme, called HIND2, from Haemophilus influenzae bacteria. This enzyme could cut DNA at specific recognition sites, a groundbreaking discovery because it allowed scientists to cut DNA precisely, not randomly. Soon after, Daniel Nathans in 1971 used Smith's enzyme to cut and map viral DNA, demonstrating how restriction enzymes could be used for genetic analysis and engineering. This practical application helped launch the era of molecular cloning. Next up is Stanley Cohen and Herbert Boyer, 1973, discovered how to insert DNA fragments into another organism's DNA. They used restriction enzymes from Smith's earlier work to cut DNA and then used another enzyme called DNA ligase to join or insert a foreign DNA fragment into a plasmid, a small circular DNA molecule found in bacteria. When this recombinant plasmid was introduced into E. coli bacteria, the bacteria replicated it, marking the first successful recombinant DNA experiment. Meganucleases were first discovered in the 1980s, not by a single scientist, but by several molecular biologists studying homing endonucleases, a type of enzyme that recognizes long DNA sequences. Bernard Duhon, 1985, discovered the first homing endonuclease, one cell in yeast mitochondria in 1985. Thierry Ducatchu, 1990s to 2000s. Thierry Ducatchu and his team at Selectis from France later developed engineered meganucleases in the 1990s to 2000s were used in gene targeting and editing in higher organisms. Zinc finger nucleases (ZFNs) were developed in the 1990s by researchers Carlos F. Barbas III, Dana Carroll, and Jeffrey Miller. These scientists engineered zinc finger proteins, originally known as DNA binding motifs, and fused them with the focal nucleus domain, allowing them to cut DNA at specific sites. Transcription activator like effector nucleases, late 2000s, developed by researchers at the University of Minnesota, notably by Adam J. Bogdanov and Daniel Voitas. Talents are built by fusing two components, a DNA binding, derived from transcription activator-like effectors, found in Xanthomonas bacteria, and the FOC1 nucleus domain that cuts the DNA.
So, gene editing is a process that lets scientists change an organism's DNA accurately. It works like cutting and fixing parts of a sentence. They can add, remove, or change specific genes to get the traits they want or fix problems that cause diseases. This technology is also used in medicine to repair damaged tissues and create healthy cells, which helps in treating illnesses. While gene gun is a tool used to insert new DNA into cells. It works by shooting tiny particles coated with genetic materials into the cells at high speed. This method is often used in genetic engineering and biotechnology to change the genes of plants, fungi, or bacteria, helping scientists create, improve, or modified organisms. And We have here the two alternative gene delivery technique. First is electroporation. Electroporation is a technique where scientists use small electric pulses to open tiny holes in a cell's membrane. These holes allow DNA, proteins, or other useful materials to enter the cell. Once the material gets inside, the cell membrane closes again. This method is commonly used to deliver genes or medicines into cells for research or treatment. Second is CRISPR-Cas9. CRISPR-Cas9 is a gene editing tool that works like precise molecular scissors. It has two main parts: the Cas9 enzyme, which cuts the DNA, and the guide RNA, which directs Cas9 to the exact spot that needs editing. After the DNA is cut, the cell naturally repairs itself, allowing scientists to add, remove, or change specific genes. This makes CRISPR-Cas9 a powerful method for studying genes and treating genetic diseases.
Good day everyone, my name is Cyrus Junokora and I'm going to discuss the key products. First is the CRISPR-Cas9 edited rice in China. We know that rice is always attacked by a pathogen, or also known as rice blast. That's the reason why scientists used CRISPR-Cas9 system. CRISPR-Cas9 system, researchers improve rice yield, disease resistance, and nutritional value. One of the important results of this study is the discovery of the lesion mimic mutant, or the LMM. This LMM will develop a black spot even there is no presence of pathogen. That is the reason why scientists used CRISPR-Cas9 to cut those specific genes that create black spot and also edit those genes that suppresses defense to make rice resistant to rice blast. Next is the non-browning mushroom in USA. These mushrooms were edited using CRISPR-Cas9 to deactivate a gene that causes browning. This innovation helps extend shelf life and reduce food waste. They use CRISPR-Cas9 to cut or shut down the production of specific enzymes that causes mushroom to turn brown. This will help not only to reduce pesticide use but also improve food quality and health. Next is the BT cotton and BT corn. This is a worldwide. Using gene gun technology, scientists inserted the Bacillus thuringiensis, or the BT gene, into corn and cotton plants. This gene produces a protein toxic to certain pests, drastically reducing pesticide use and increasing yields. It produces toxic proteins, or the cryptoproteins, and once the insect pests eat the plant, it will lead them to cause death. But it is not harmful in humans. Next is the golden rice in Philippines. It is developed through genetic engineering. Golden rice produces beta-carotene, a source of vitamin A to combat deficiency in developing countries. This beta-carotene is converted into vitamin A by the human body. Golden rice helps combat malnutrition and childhood blindness caused by vitamin A deficient. Next is the PEF, or pulse electric fields used apple in Ireland. PEF can be used to treat hard apples after harvest, perforating their cells and increasing the efficiency of juice extraction. In Ireland, PEF technology is widely studied and applied in apple juice industry. Researchers found that PEF increases juice yield, preserves flavor, and extends shelf life without using heat. This makes apple processing more sustainable and energy efficient while keeping the natural quality of the fruit. Irish food companies are now adopting commercial PEF systems to produce premium quality apple juice for export.
So let's proceed to importance and applications of gene editing. So in disease-resistant plant crops, this helps protect the plants from diseases such as bacteria, fungi, and viruses. So farmers can grow better crops with fewer losses. So in drought and climate-resilient crop, in relation to climate change, CRISPR-Cas9 helps create the crops to survive drought, heat, and other effects of climate change. Other than CRISPR-Cas9, we have also a tool like MAB, or the Mark-Assisted Breeding, which uses DNA to identify genes for drought or other effects for climate change. So let's proceed to technical drawbacks. First is the off-target effects. This happens when the gene editing accidentally changes the wrong part of the DNA, causing the wrong or the unwanted mutations. Second is the delivery problems. It occurs when it's hard to insert gene editing tools to the tissues or the cells of the organisms. The third one, or the last one, is the incomplete editing. Means not all cells are changed, resulting in a mix of edited and unedited cells called mycelium.
Good day everyone, I am Janet Paglinawan, and the countries nga nag-approve of genetically modified crops kay include na America, Philippines, Kenya, Canada, Bangladesh, Brazil, and many more. So some total of the countries nga approved is 23 countries. Updated na siya sa year 2024. So, sa year 2020, ang Kenya, they announced the approval of BT corn after 5 years field trial exhibited effects to African bollworm and increased their yield. So, sa East Africa, Nigeria, and other West African countries is nag-approve sila sa virus-resistant cassava varieties. Sa Indonesia naman is nag-start sila conducting for safety studies for both granola and diamond varieties of GM potatoes with stock genes. Sa Costa Rica naman is ilang researchers are working on drought-resistant rice and GM pink pineapple with higher level of lycopene. So more countries are adapting to GMO crops because of its contribution to food security, sustainability, and climate change solutions. It increases crop productivity, it conserves biodiversity, and it provides better environment. Also, nag-reduce po siya CO2 emissions, and it helps alleviate poverty by uplifting the economic situation. So, naay mga countries nga wala nila gi-approve ang pag-cultivate and ang pag-import og GMO crops because of these factors. Number one is trade protectionism. Ilang proteksyonan ang mga ilang domestic producers of foods, and second is pressure sa mga activists, itong mga people nga mga consumer nga conscious sila sa ilang mga ginakaon, so dili sila mug-buy og mga products nga na genetically modified. And third kay public uneasiness or a desire to protect the country's image, such as the French belief that ang genetic crops could contaminate the country's reputation as a world food capital. So most countries nga wala nag-approve aning GMO crops kay ang mga European Union countries because of their image na kanang as the world food capital.